a comparison of the influence of two different mesh materials on the foreign body reaction. All Dynamesh implants are primarily made of polyvinylidene fluoride, or PVDF for short. This material has good biocompatibility, reducing the foreign body reaction compared to other materials such as polypropylene. In the following film, we will demonstrate important pathophysiological processes and compare the influence of a mesh material with a low and a strong foreign body reaction. Have you ever wondered what actually happens when a mesh is implanted? How does the tissue react to the polymer and the structure? Imagine you were standing on the surface of the mesh implant and could observe what happens here a few milliseconds after the implantation. Almost immediately after implantation of the mesh, the tissue reacts to the foreign body. Autologous proteins such as albumin and fibrinogen are adsorbed to the surface. The changes to and denaturation of the proteins represent an immunological stimulus, in particular for the macrophage, which can be found early on at the material-tissue interface. The macrophage is activated as a result of the contact with the proteins and, in turn, releases cytokines that initiate the foreign body reaction. More and more macrophages are chemotactically attracted to the site where they are involved in the early granuloma formation. After just a few hours, the mesh implant becomes enveloped and covered by these macrophages. During the release of the cytokines, other important cells like fibroblasts are attracted, which produce the collagen of the fibrotic capsule that gradually shields the mesh implant. In fact, numerous fibroblasts are already present on the first day, forming an early perifilamentary scar. With time, T lymphocytes are also attracted. Occasionally, isolated polymorphonuclear granulocytes are still attracted to the implant site, which disappear as the granuloma ages. As the process continues, the number of cells in the granuloma increases. After 1 week, an early foreign body granuloma with the typical layering of an inner cellular infiltrate rich in macrophages and an outer fibrotic capsule of collagen and fibroblasts can be recognized. The foreign body granuloma now begins to mature, and increased vascularization becomes noticeable after about 2 weeks. The endothelial cells initially form simple endothelial tubes, which later develop into capillaries. The extent of the foreign body reaction and the extent to which the granuloma envelops the individual mesh fibers are essentially dependent on the polymer used. The extent of the outer fibrotic capsule is particularly important for the subsequent function of the mesh implant. A material that elicits a low foreign body reaction, for example, due to low stimulation of macrophages, results in a smaller granuloma with its fibrotic capsule. In contrast, a material that evokes a strong foreign body reaction results in a more pronounced cell layer and peripheral tissue capsule. Let us now leave the surface of the mesh implant and look at the implant as a whole from above. After implantation, the mesh implant is quickly covered with proteins and the foreign body granuloma starts to form. The connective tissue capsule is shown here in purple. Here is a schematic cross-section of the foreign body granuloma. The mesh fibers are shown in white, the adsorbed proteins in brown, the cellular infiltrate in pink, and the outer fibrotic capsule in purple. Now, let us compare a material with a low foreign body reaction to one with a strong foreign body reaction. With the same distance between adjacent fibers and the same thickness, the connective tissue capsule shown here in purple is smaller for the material with a low foreign body reaction. This means there is a lower risk of bridging and excessive connective tissue formation for a material with a strong foreign body reaction. This means the open pore structure is essentially reduced. In contrast, a material with a low foreign body reaction retains its open pore structure much more effectively.